Hello, everybody. My name is Nicole Boos, and today we will be learning about rabies. What is rabies? Okay, so rabies is classified within the Rhabdoviridae family. It's an enveloped virus with a helical capsid structure. It's bullet-shaped and approximately 170 nanometers long and 80 nanometers wide. Disease description. Areas infected. Rabies is characterized by pathological changes that are primarily in the brain and spinal cord. It enters via bite site and spreads through the nerves, causing inflammation and neuronal damage. Signs and symptoms. Early signs and symptoms include flu-like and or irritation at bite site. Also, things like fever, pain and itching around the bite area, headache, maybe some insomnia. Progressed symptoms would include neurological symptoms and muscle spasms, aggression, hallucinations, partial paralysis, light sensitivity. And some severe signs and symptoms include hydrophobia or aerophobia, which would be fear of drinking water or fear of drafts of air. Incubation period is typically 20 to 90 days. There have been cases as short as 4 days and as long as 6 years documented. Disease frequency, specific regions. This includes Asia and Africa, where resources are limited in preventing rabies. Susceptible individuals include veterinarians and animal handlers, also wildlife biologists or researchers, cave explorers, campers, hikers, children, people with certain weakened immune systems, and also people living in resource limited areas like developing countries. Morbidity and mortality. Rabies is 100% fatal once clinical symptoms appear. There's 70,000 annual deaths worldwide attributed to rabies. Most of these occur in Asia and Africa. And dogs are responsible for 99% of human rabies deaths outside of the US. Source. Where does rabies live? Rabies is zoonotic, which means it's a disease that can be transmitted from animals to humans. Where does rabies come from? Rabies can come from animals like bats, raccoons, foxes, and unvaccinated dogs. Also, coyotes, skunks, and unvaccinated livestock as well. Transmission methods. Most common methods include bites and scratches from infected animals, and less common methods include saliva from infected animal entering through a cut or coming in contact with mucous membranes. Prevention. Avoiding exposure or contact with wild animals. Key measures include vaccinating pets and getting prompt medical care if exposed. Therapy. What are the treatments and vaccines for rabies? There is pre-exposure prophylaxis and post-exposure prophylaxis, both known as PEP. A Approximately 16,000 to 39,000 people come into contact with potentially rabid animals and receive PEP each year. This includes wound care, HRIG, which is human rabies immunoglobulin, and this provides an immediate passive immunity by supplying virus neutralizing antibodies that bind to rabies virus at the wound site, preventing its entry into the nervous system. And it's given as soon as possible after exposure. And rabies vaccines that stimulate the body to produce its own virus neutralizing antibodies through immune recognition and memory production is also given. This triggers both B and T cell responses against the rabies virus, glycoprotein, and it enables the immune system to recognize the virus and clear it. Vaccines are initiated promptly after exposure on a multi-dose schedule. And for pre-exposure prophylaxis, vaccines are given as a series before potential exposure. Immunity. Since rabies is usually 100% fatal once infection sets in, infection itself does not mean lasting immunity, but vaccination can provide lasting and potentially years-long immunity, especially when booster doses are given as recommended. PEP is highly effective and, when given promptly and correctly, essentially prevents rabies death. Tests. How is rabies diagnosed? One test is the direct fluorescent antibody test, DFAT, and it's the standard for rabies diagnosis post-mortem. It is performed on brain tissue and detects rabies virus antigens using fluorescently labeled antibodies. There's also reverse transcriptase polymerase chain reaction, PT-PCR. It's used to detect rabies virus RNA in saliva, CSF, skin biopsies, or brain tissue. It's useful for both antemortem and postmortem diagnosis. There's also virus isolation, which is performed through clinical samples from saliva, CSF, tissue, things like that. And it uses cell cultures or live mice to confirm rabies virus. There's a antibody detection, which is blood tests for rabies virus neutralizing antibodies, which is useful for confirming immune status or response to vaccination, but not for diagnosing acute infection. There's also histopathology and immunohistochemistry. Postmortem histology is used to identify Negri bodies in neural tissue, and immunohistochemistry uses labeled antibodies to detect viral antigen in tissue sections. Some new data on rabies include recent large-scale analysis reveals that immunological and molecular tests provide better accuracy for rabies diagnosis than the long-standing DFAT test. Also, rapid field tests improve rabies control and save lives by allowing early intervention, reducing transmission, and decreasing mortality rates, especially in endemic regions. And here is my sources. And that's it. Thank you. Hope you learned a lot.